this for those who are not aware. So if you would prefer to not be recorded, then perhaps keep your video off throughout the entirety of the presentation. And if you have any questions, then by all means, type them out in the chat box. My coworker Sabrina Weber, who's on our SCTLD response team as well, is going to be here as a moderator / co-host. So she will be keeping track of the attendance, those who are joining us, and also keeping track of all the questions. And we're going to kind of split this up into two sections. So I'll be presenting the first section on coral biology and ecology, and then we'll have a short 10-15 minute break. And then Sabrina will take over for the coral species identification and the different levels of susceptibility for SCTLD that we have in those species of corals. So you can have questions for the first half with me, and then we'll kind of trade and do questions for the second half. Okay. So I don't know if I've introduced myself. I'm clearly not Dr. Croy McCoy. My name is Katie Ebanks, and I'm one of the SCTLD response divers here at the Department of Environment of the Cayman Islands. So thank you to everyone for joining us today. Really appreciate it. And again, this will be recorded and be posted on our Cayman Islands DoE YouTube channel so that you can review it at another time, or so that other volunteers can watch them when they have the time to join us. Okay. So for today's presentation, we're going to have some learning outcomes in two sections. First, we're going to have coral biology and ecology. Then we're going to talk about some different key distinguishing characteristics of stony corals versus other species of coral, and how to use certain key words in being able to describe one coral species from another in the field, or when you come back from the field and you're analyzing photos and things like that. Then we're going to talk about the different stony coral tissue loss disease classification groups. So there are over 47 different species of stony corals in the Caribbean alone. However, not all of them are susceptible to SCTLD that we know of at this point. So we kind of divide those into three groups. So we'll go into those three groups. Then we'll have a 10-15 minute break for everyone to have some water, use the restroom, just stretch your legs because, you know, Zoom meetings. And then we'll go into Sabrina's section with some coral identification and the indicators of SCTLD on those species, how we can tell and how you can tell as one of our volunteer response divers, what disease is it? Is it SCTLD, or is it one of the diseases that you perhaps learned about yesterday with Dr. Croy McCoy, like a white plague or a black band or something like that? Okay, so as I said, there are different kinds of Caribbean corals. There are stony corals, also known as hermatypic species. These are your reef building corals. These are corals that have a calcium carbonate and aragonite skeletal structure, which even when the corals die, will leave behind this skeletal structure and kind of build onto the reef structure, creating that really nice 3D framework on the reef that we all know and love so much today. There's also soft corals or gorgonians. These are your sea fans, your sea rods, the really beautiful corals that are kind of like waving in the you know, the current, and they're still corals, but they're not producing that skeletal structure to leave behind for other creatures to build up on, creating that complex reef framework. So still an important species, however, not a reef building species. Then there's hydrocorals. I'm sure as many of you on this call are very experienced divers, we've all had a run-in or two with some of the hydrozoans or the hydrocorals, which are your fire corals and your lace corals. So on the bottom here, if you can see my cursor, this is a lace coral or rose coral, and you see that in a lot of cave systems. But again, these species of hydrocorals are not contributing a calcium carbonate deposit behind when they continue growing or once they perish and other corals come on top of them. These don't leave that behind to build a reef structure upon. And then you have your black corals, also known as your deep water corals. If hopefully everybody on this call is an advanced diver, or you're working towards your advanced diver certification, in which case you've done some deep diving, still within the recreational dive limits, of course, but once you get down to about 80 to 100 feet, then you're going to start to see some really large, what look like soft corals, but they're a lot thinner and they're really big, and they're usually on the wall in the Cayman Islands. And those are your black corals, or your deep water corals. But today, for this presentation, as well as for the entire stony coral tissue loss disease project, we're going to be focusing on those stony corals, or the hermatypic species of corals, because those are the corals that scientists know of today that are susceptible to the stony coral tissue loss disease, hence the name, stony coral tissue loss disease. So that's what we're going to be focusing on today. So at its most basic, what is a coral? Before getting into marine science and marine biology, I have to say that I wasn't totally sure what a coral was, and we hear all the time, I'm sure many of you on this call have as well, what is a coral? Is it an animal? Is it a rock? Is it an algae? Like, what is this squishy creature on the reef that I'm not supposed to touch? So basically, a coral is all three. It is first and foremost a colony of creatures working together. So in the top left corner, you've got a Montessori cavernosa species, or the great star coral. If you've dove on the reefs of Grand Cayman or in the Cayman Islands, you've probably seen these quite frequently. It has really large polyps, about 2 to 3 centimeters sometimes in diameter, over an inch occasionally, depending if you're using metric or imperial. And those polyps are quite large, and all together, they're creating this colony, this mass of many different polyps, all working together as one organism. And it kind of, like I said, has all of those things together. A coral is an animal, but it is also housing algae, or a plant, and then it's depositing that skeletal structure, creating a rock left behind. So in this top left photo, you see all of these polyps all together on this colony. It's quite a large colony. And then as you look a little bit closer into the individual, you get the individual polyps, which look like many different tiny little anemones. And each of those anemones has those tentacles and is able to move those tentacles to grab passing food. Passing food can be detritus falling through the ocean, the water column. It could be small fishes, it could be bacteria, it could be dinoflagellates, whatever small microscopic creatures happen to be kind of falling onto that coral. But also, that's not the way they achieve most of their nutrients and most of their energy. So how they do that is inside of each individual polyp. You can see in this top right photo here, there's some little greenish brown specks. And sometimes you can see these with the naked eye, but usually you'll need a microscope, which brings it to the lower right-hand photo, which is its zooxanthellae. So the zooxanthellae is a photosynthetic algae. It's a single-celled organism which is living inside of the coral host, and this zooxanthellae is converting all of the sun's energy into food and nutrients for the entire coral colony through photosynthesis. So that is how the coral animal, the entire colony, obtains most of its nutrients, is through that photosynthesis and through that symbiotic relationship with the zooxanthellae. However, 5 to 10%, depending on what species of coral, are able to grab with their tentacles for passing by food. So a coral is a little bit of everything, really. How can we identify corals from everything else on the reef? Most divers, when you first start out, obviously you notice the big stuff first, the sharks, the whales, the dolphins, the fish. And then as you start diving more and more, you start to kind of focus your large field of view narrower and narrower and narrower onto the small creatures. And kind of the details of what makes up all of the life on this coral reef. And that's when you start to really notice that all of that stony stuff actually is very different. 47 different species of stony coral on the Caribbean reefs alone. That's not including the hundreds of species of sea pens, gorgonians, or soft corals, and things like that. And that's not even touching on all the other reef creatures, algae and tunicates and bryozoans and all kinds of things. So how can we tell which ones are corals and which ones are other animals or other creatures? Well, several ways. You can identify a coral by the size of the colony, what depth that colony is in, the potential coral that you're seeing. Is it super shallow? Is it super deep? Where in the reef system is this coral? Looking at the coral-like cup structure, which is the kind of thing that's holding that polyp in place, that part of the skeletal structure of the coral, which is housing that little baby anemone or that little polyp. So that structure looks completely different on many different species of coral. Some can be described as really large, like that great star coral can be, up to an inch or several centimeters in diameter. They can be really tiny, like less than a millimeter in size. Some are what you'd call innies. So when you're teaching kids about corals, they're like innies and outies belly buttons. So some look like you could poke them with a pencil and it left behind this little mark, and that's the innie. And some coral-like cups stick up from the surface of the colony, and that's an outie polyp. So many different ways of looking at how they are across the colony of the coral. Are they smooth across the coral? Are they indented, or do they raise up a little bit? And that's what I mean with flush with the colony, or are they protruding? And then looking at the color of the coral. This is unfortunately not a very great way of determining a species of coral, because many of the corals are the same color. So you can't just say, oh, I found a Paredes asteroides and it was a kind of rich gold color. And it's like, well, there are many rich gold color corals out there. So this is a great indicator in addition to these other key distinguishing characteristics, but it's not great as the only thing to describe the coral colony that you're looking at. And as one of our volunteer divers, if you're trying to describe to us one of the colonies you treated, this will help, but we'll need some more information. Looking at the ridges, valleys, and the grooves. So brain corals, I feel like, are one of the most difficult things to start to identify within the morphology of brain corals, one species from the other. But we can start to talk about how high are the ridges, how deep is that groove, and actually, does that ridge have a groove inside of itself? All of these little attention to detail key distinguishing characteristics will really help you to identify these species. Of course, we'll talk about morphologies, if they're plating corals, branching species, big giant boulder species. We'll go over all of that. And then, as I said, corals are a colony of polyps all working together, but occasionally there are some species of coral that is solitary. It's just one polyp makes up the entire animal. And there are a few like that, so we'll go over those. Okay, so an example of some coral identifiers. We've got small colonies, we've got medium colonies, and we have large colonies. I apologize, there's no scale bar in this presentation, but take my word for it. These species are small, medium, and large. The small coral, again, this is kind of up to you as the volunteer diver, right? What do you consider small? I suppose for me, I would consider small anything maybe smaller than a melon. Okay, that's kind of small. And then for medium, I would say melon size to maybe beach ball size. And then anything bigger than a beach ball size, I would say is large. So that coral colony down in the lower left-hand corner, that's an Orbicella annularis, or a lobe star coral. And difficult to tell from this photo, but that's probably about 2 meters across, or 6 feet or more across in diameter. So, if it's a U-shaped coral, it's large, and you can kind of go from there in your description. And then what depth is that coral in? Is it shallow on the reef? Maybe, above 30 feet, 10 meters, pretty shallow species. Is it intermediate? You found it on one of the spur and groove formations, and it was between 30 feet and 60 feet, or is it below 60 or 80 feet and it's kind of going off that deep wall, like we talked about the black corals? So where are you seeing this colony of coral that you're trying to identify? Shallow, intermediate, and deep. And you could even include what type of habitat it's in. So are you on the hard bottom substrate, kind of on your way out to one of the shore diving sites where there's not a whole lot going on in there? We will see some stony corals in there, but they're quite small usually, and they blend in with the bottom very well. And then, did you see it on, like I said, the spur and groove reef formation? Did you see it over the wall? Did you see it on a patch reef? These are things, describing the site where you saw the coral will help us to determine what species it is, species A or species B. And then talking a little more about those innies or outies and the coralite cup structure. On this slide, you see some skeletons of various species of coral, all of which belong here in the Caribbean. So these are all coral skeletons that if you walk along the beach and you see some rubble rolling around, pick it up and look at it. It's probably a coral, and you can tell a lot by looking at the coral species when you see its skeleton. So I encourage you to pick those up and then, of course, to put them back and take a look at it. Does it look like the lower left picture down here where it's kind of wide coralite cup structure? You can see the lines in between it, which are called the septa. So you can see those individual coral teeth or septa inside of those little grooves. Or is it like the second photo from the left up here where someone's holding what looks like it could be Cyderastria or the greater starlet coral, where if you look at it from far away underwater, they're quite rounded and they look uniform and kind of smooth. But then when you go up to it closer, it's almost like you've rolled up a ball of putty and then poked it a million times with a pencil or a pen. So lots of small indentations. And the one on the third photo from the left is, I think, the same thing, Cyderastria sidera, but very, very close, where you can actually see those small holes where the polyp would be housed, but they may only be a millimeter across in diameter, or less. It's quite small and up close. And then the last photo in the upper right is from a brain coral species. So you can see, even though it's just the skeleton, you can see those ridges and those grooves as well. Okay. And so here's some living tissue colonies, close-ups of those coralite cups or those polyps and that structure. So what is that living tissue layer on top of the skeleton structure look like, and what shape does it give the coral overall? So here we've got a great star coral again. Very famous species. We're using a lot of examples of that. And it looks almost fleshy. We typically, as I'm sure you all know on this training session, we typically will not touch corals. You don't touch anything on the reef. You leave it as is, take photos, but leave only footprints, that sort of thing. With the SCTLD response project, it is likely you'll be touching corals, especially when you are applying the antibiotic mixture. But of course, we ask you to be very ginger about this, apply it very, very gently. We ask you to have perfect buoyancy so your fins aren't hitting the reef anywhere and your other hand that you're not using is not flailing around. So you're being very gentle when you're applying it. But you will see that some appear fleshier than others. And this is one of those species that it looks like if you were going to touch it, it might be squishy, but it is a stony coral as well, and it's not squishy. But this is one of the more difficult ones to apply antibiotics to, which you may have heard in Jay's presentation yesterday. And if not, then you will definitely get some hands-on experience with that when you start to come out with us. Here's an up-close photo of one of the brain corals. And this is what you're talking about with the ridges here and then the deep grooves here. I believe this, it's hard to tell with scale, but off of this photo, I would say it is a symmetrical brain coral or potentially a giant brain coral. So hard to see here based on the photo, but it has these large ridges that come up pretty high off the skeletal structure. And then these ridges, or sorry, these grooves, which look pretty deep. So that is a key distinguishing characteristic describing how far away the ridge and the groove are, or how widely spaced they are. So on the Boulder brain coral, the Copophilian natans, these ridges can be inches apart from each other. But on the symmetrical brain coral, the Pseudopluria strigosa, they can be very, very close to each other. That's why it might be hard to discern based on this photo without scale which of those species it is exactly, but you can start to get a feel for the differences in the brain corals by looking at these up close photos. And then this is the miandrina meandrites, the maze coral. And you can kind of see it looks similar to the last photo, which was a symmetrical brain or a boulder brain coral, in that this one, it still has these ridges, right? And it has these grooves, but the ridges look a little different. I've heard people also call this the zipper coral, like it looks like you could put a little zipper on there and almost zip the whole thing up, and that's one way to remember it. But you can almost see plates stacked up against each other. So these septa on the corallite cup structure are really pronounced, whereas in the previous brain coral, they weren't as pronounced as this. It's almost like somebody stacked a bunch of discs on top of each other and created this ridge. And the groove is not nearly as deep and it's not as wide. So already, maybe you don't know what this species is, but you can start to see that there are differences in what you would think this is a brain coral and what the previous one was as a true brain coral. And then here is one of my favorite species. This is a Musa angulosa, and this is a kind of mix between a solitary colony and a true colonial species of coral, where each of these polyps is massive. They can get up to 6 to 8 inches in diameter each, so quite large. And they grow in almost like a bunch, similar to a bouquet of flowers that kind of have stalks that all keep the colony attached as one, but then they grow up and the living section of tissue is just the top portion where like the flower would be. And that's what each of these polyps, these giant polyps, is representing. It's one of those kind of flower tops. And you will see these occasionally on the reef as well. Its key distinguishing characteristic is, again, the fact that they're massive, much larger polyps than any other species that we see on our reef. So a really great key distinguishing characteristic. And then this is a up-close photo of many of the boulder colonies that you'll see while diving. So the Montessorias, the Orbicellas, these are some of the star corals. So the mountainous star coral, the lobe star coral, a lot of them will have polyps that look identical to this. Up close, they appear very like they're coming off of the structure quite a bit, like they're very high, but from far away, they're so small that it appears very smooth, and you really can't see the individual polyps or corallite cups very well. So this is a very close-up photo of one of those. Okay, we've sort of already seen a few of the morphological differences just from those photos of solitary colonies versus colonial colonies. But here are a few more morphologies that you'll see corals being described as. You've got your flower cup corals here on the upper left. Below that, you have your branching and pillar corals. These are some of the favorites for photographers. And if you're familiar with any coral nursery programs, this is typically the species of coral that people are growing within their coral nurseries because they are the fastest growing species, both on the reef and in a nursery setting. And then you have your encrusting, mounding, and boulder corals. So these are the really large corals that you're seeing on the reef. Below that, you've got plating or leaf and sheet corals, and these can be seen just as you see in this this drawing or this sketch, where they're just laying on top of the reef and kind of growing outwards and upwards. Or most often in the Cayman Islands, you'll see these kind of off the wall, sheeting on the edge of the wall and kind of bringing a little lip out, so they get a little bit of that sun for their zooxanthellae to go through photosynthesis for them. And then the upper right-hand corner, of course, you've got your brain corals, and below that, your fleshy corals. Okay, so what is a coral with SCTLD? How do we determine what is a coral colony with SCTLD versus one that does not have SCTLD? So here you've got your coral colony again, our Montessori cavernosa, or great star coral. And this is a coral infected, visibly infected with stony coral tissue loss disease. So you can see that right here, there's a stark white margin, and then the brown is all of the living tissue of the coral colony. I'm sure you saw many photos similar to this in Dr. Croy McCoy's presentation yesterday, but this is what a coral colony infected with SCTLD looks like. Many different ways that SCTLD is expressed depending on the species of coral that it's affecting, and it can be very irregular in its shape. It's not a band, it's not a blotch, it's not a dot, it's not many dots. It's very irregular, as I'm sure you've already heard. And here's another example, a little bit closer up, of the polyps. Here you can see that, you know, this polyp down here is actually shedding its tissue, you know, in the moment of this photo, what we call sloughing the tissue. If you were down there and you wafted it a little bit with your hand, bits of that tissue would actually be coming apart from the coral structure. And so on certain species, like the great star coral, you can actually see this with your naked eye underwater, because it's so distinct, that tissue coming off that skeletal structure. And so what does a coral with SCTLD look like to the individual polyp? That's a good question. We don't really know. You know, we've seen it affecting individual polyps, like down here on the Montessori cavernosa, but what's happening inside of that polyp? What's happening inside the zooxanthellae? What's going on with that relationship, and how is it affecting that relationship? Unfortunately, at this time, we really don't know. There's a lot of scientific research going on about this all over the Caribbean and the Florida region of the United States. Unfortunately, nothing is really known too much yet. We do know that the antibiotics are helping, and that's why you're all interested in helping us today, right? Is to help the reef and help the corals as much as we can with the latest updates, information from the scientific groups. And so, as stated, we're going to go through different levels of susceptibility from species of coral to stony coral tissue loss disease. So here you've got the 8 highly susceptible species that you might see here in the Cayman Islands. Again, there may be more or less on this list dependent on the region where the disease is present. So if somebody is coming to us after volunteering with a SCTLD response team in Florida or in the Virgin Islands, this may look like a very different list than what we have here in Cayman. So we've got the smooth flower coral up here on the top left. That's a highly susceptible species. This is typically a small coral, rich gold in color. Sabrina will go more in depth on that. You've also got next to it the elliptical star coral, or Dichoscinia stokesy, again, highly susceptible species. These top 4 really are the 4 that here in Cayman are the first to get infected, and usually the infection spreads very quickly. Got the maize coral, the pillar coral, unfortunately, the very famous giant pillar coral colonies. Thank goodness we've got an East Coast strike team out of Ocean Frontiers on the east end of Grand Cayman, where the majority of Grand Cayman's pillar coral colonies are located, and they're making sure those colonies are getting extra special attention with treatments all the time. And then you've got your symmetrical brain coral on the bottom, your knobby brain coral, your boulder brain coral, and your grooved brain coral. So unfortunately, the brain corals are getting hit pretty hard with this disease. For your intermediate susceptibility species, some of the larger mountainous and boulder star corals. So you've got the lobe star coral up here in the top left, and then you've got the mountainous star coral next to it and the boulder star coral. All of those, the Montessori's and the Orbicellids, pretty susceptible to SCTLD. Not as bad as the first slide, but we do treat quite a bit of these corals on the reefs of Grand Cayman. On the bottom line, you've got the great star coral, which is the one we keep using examples of in this presentation. Then you've got that massive starlet coral and some of the lettuce corals. Lettuce corals also can look like the plating species. So the ones that I was saying, if you go off the wall, kind of grow on the side of the wall and will grow a little bit out to get some of that sunlight access. Many different morphologies of that lettuce coral on the reef. So we do treat quite a bit of that as well. And then some low susceptibility species. Thank goodness, the staghorn coral and the elkhorn coral on the bottom row here are a low susceptibility species. They are some of the most popular, like I said, for nurseries. So there are quite a few nurseries in the Cayman Islands that are housing staghorn coral, elkhorn coral, and now some are even housing the pillar coral. So, we're very thankful this disease so far does not seem to be affecting that genus, which is the Acropora genus. But again, this is just based on knowledge that is constantly ongoing. And the top row is mustard hill coral and your finger coral. So these are corals that are in the Porites genus. Another branching species, but they're kind of stubby and they stay closer to the reef. We have not yet observed SCTLD on any of these species. But of course, as I said, everything is ongoing. So as of right now, low susceptibility to no susceptibility to SCTLD. But as with nature, she doesn't love rules. So that's just for now, and we'll continue to monitor that. So the whole reason that those of you are joining us on this training today and have done so yesterday and last week is because you're really and truly interested about helping the reefs of the Cayman Islands. You're passionate about the beauty that is the reef and hopefully want to see it around for a long time. And this disease is upsetting for you reasonably. So we really need your help. We have a dedicated team here at the Department of Environment of about 10 people whose sole purpose is working with this disease, treating corals on the reef, looking at the disease outbreaks around the island, towing around and trying to see where the new edge is, and trying to learn as much as we possibly can about the disease from other stakeholders in the region who are also conducting research so that we can learn more lessons and know how to respond and how to mitigate the unfortunate spread that is this disease. And so thankfully you're interested, and we can use all the extra help and volunteers that we can get. We can get multiple boats out on the water. If we can get more people on each of those boats, if we can start doing some more outreach events, all of that helps. And so there are 10 of us, which is incredible, but it takes even more people than that to make this project happen. And I believe you saw this slide yesterday, but just identifying stony coral tissue loss disease on a colony that's actively fighting this infection. You can see this arrow on the brown tissue. This is living part of the coral colony that is visibly unaffected as of when this photo was taken. The middle arrow is pointing to the bright white edge, stark white, bare skeleton. This is an area, this is what we call the disease margin, where the disease is interacting with the live tissue and killing it off pretty rapidly, and so rapidly that it's leaving behind just a bare white skeleton. And then the lower arrow is showing kind of a greener, browner, not so stark white part of the skeleton. And that's just simply showing that this is where the disease first showed up in this lesion. And then it grew outwards in an irregular looking circle from there. And so over time, it just developed some color on that skeletal structure because of small bits of algae starting to colonize that old skeletal structure, and other creatures, bioeroders, maybe even other organisms settling on it. And it starts to change the color a little bit. It's not as bright white. And so that's old dead from the disease. Okay. And we kind of talked a little bit about lesion shape earlier and how this disease, it doesn't follow any particular pattern. As you can see all the way on the left, it's quite linear. It can be a line just moving uniformly across a coral colony. It can be unifocal, like the center photo, where it just starts right in the center of a colony and grows outwards in a perfect circle from that one spot. Or it can be multifocal, like the photo on the far right, where you can see that there are probably multiple areas, multiple lesions that started here and have all kind of worked their way outwards, and now they're starting to meet each other. So again, it's very irregular, and I'm sure that you saw many photos similar to this yesterday in the disease presentation. And that's it for the basic biology and ecology portion of this presentation. It is 2:40, so why don't we take a few minutes for questions and then have a little break. So does anybody have any questions just about that first portion of the presentation about the coral ecology or biology? And I see that Sabrina is messaging you guys if you end up having any questions related to anatomy of corals or their biology or anything like that, feel free just to post it in the chat and we can get to it another time. Okay. Besides that, why don't we take a 10-minute break and Sabrina and I will swap presenter stations and we can come back at, let's say 10 to 3, so 10 minutes from now. Okay,
Thanks.
Thanks everyone. I'm going to go ahead and turn off the video for each of us just for a few minutes. We're going to remain connected, so feel free again to post those questions, but we will be turning the video off just for a few minutes. Okay.
Okay. Okay, so here's the susceptibility key. So Katie touched a little bit on the susceptibility of some of the corals that we're going to be looking at. So if it's in pink, they're highly susceptible corals. The darker yellow or the orange is moderate susceptibility, and the bright yellow is low susceptibility. So we talked about a few of those corals, but I'm going to be going into more depth on them. So they do, different species differ in their susceptibility to SCTLD, and some species do appear to be immune to its effect or have very low susceptibility. So this is also one of the ways that we know how long SCTLD has been on a site. We can't know for sure sometimes, but I feel like part of our job sometimes we're doing a bit of detective work. So especially when we're trying to find some of these leading edge areas, we look for the highly susceptible corals in sites where we think there may potentially be SCTLD. So I'll go over some of those first. Okay, so we touched on this one a little bit, but Dendrodon cylindress, pillar coral. So this is one of our endangered corals here. It's a very beautiful coral, and you're very lucky if you see this coral, especially on a frequent basis. There's less than 20 colonies in the 3 of the Cayman Islands. So we're really trying to preserve this coral. So as Katie mentioned, we have an East End strike team, and part of their priority is to treat this coral. Unfortunately, they're very susceptible to SCTLD. So they have polyps that are extended during the daytime, so they almost have like a fuzzy appearance, and this is potentially the reason that they're one of the most susceptible corals to SCTLD. That's what some people are thinking, because their polyps are always extended in the day, ready to receive any infection coming around, any of the mucus sloughing off of the other corals, but it's hard to know. So these ones, I call them carpeted tower corals. You have these big cylinders, so they're up to 10 feet tall, and their pillar diameters can be 3 to 5 inches. They can be really huge. They span 4 to 65 feet. Their family is Miandrinidae, and their acronym is DCYL. So for the acronyms, it's the first letter of the first word and the second 3 letters of the second word. So sometimes it can be difficult to remember the entire name or to pronounce the entire name. Everyone pronounces them a little bit differently, but DCYL is an easy way to recognize that as well. And most people should know what you're talking about, especially on our team. That's how we'll describe a lot of the corals, is by their acronym. So as I mentioned, the colonies form cylindrical spires that grow upward. The tentacles are extended during daytime, and it has a fuzzy appearance, and it appears to be a golden brown color. They are very beautiful to see, and most of them are out in East End in Grand Cayman. We've got our miandrina meandrites maize coral. So this is another one of our most susceptible corals. So especially if you're not out east, you may not always be seeing these DCYL corals, because they're more of a rare occurrence, but we do see a lot of miandrina meandrites. So that'll be one of our biggest distinguishers is their SCTLD that's just hit this site. A lot of the times when there's a new site with SCTLD, we'll see a lot of the mias that are dead. A lot of them are half alive. Some of them are untouched. So it's very strange sometimes, but their family is Miandrinidae as well, and their acronym is MIA. So that's a lot easier to say. So they're quite small. They can grow up to 3 foot, and they span 2 to 240 feet. They can form these flattened plates or hemispherical heads, so like almost like a bit of a boulder, or a pillar. So one that we don't discuss, we've also got Miandrina jacksoni. Difficult to distinguish between the two, and you won't have to distinguish between the two. They're the same family, but jacksoni is, they have white also between their ridges. So as Katie discussed before, they have, oops, sorry. I'm trying to move this. They have lines along the ridge tops, so it does almost look like plates, and the ridges are wide and they're very close together. So for me, I think of this coral as a bunch of fat worms. So you can think of different fun ways to distinguish between the different corals, but that's how I've always seen it. So these ridges are very wide and they're close together, and it looks like a bunch of worms to me that are just meandering through. And that's how I remember the name, Miandrina meandrites. Alrighty. And we've got Degostenia stokesy, the elliptical star coral. So again, a part of the same family. The acronym is DSTO. They're quite small, up to 1 1/4 foot. The depth is 12 to 225 feet. So some of the colonies have small coralites that barely project above the surface. So it appears to be quite flat actually. And then some have small coralites that are quite uneven and they protrude noticeably above the surface. So they seem also quite lumpy. But their coralites are quite distinguishing. They appear almost like a smiley face, I think. So that's how I think of that coral. And they're circular to elliptical, but some of them are Y-shaped or worm-like or triangular. So they have this quite funky shape, and they're all spaced a little bit apart from each other. This is one of the most susceptible species as well, and you'll see them in small colonies. Eusimia fastigiata, so the smooth flower coral. Their acronym is EFAS. They grow up to 2.5 feet. Their depth is 3 to 200 feet. And this one I think looks like a bouquet of flowers. So for this one, we actually don't treat Eusymnia fastigiata. There's not enough science on really how to treat this coral. So you'll see each of these different polyps. So in the picture below, some of those polyps appear to be completely infected or completely dead. It's stark white. Sometimes you'll see that, and rarely you'll also see maybe half the polyp that appears to be infected, half of it that appears to be healthy. So we aren't sure how to treat this coral. If you should smother the entire polyp, even if it's already dead and infected, or just because it's one connected system, we're not sure if that would even make an impact, if it would still infect its entire colony. And these die quite quickly. And again, they are quite susceptible to SCTLD. So we focus on other corals that we know have a better success rate to the antibiotics. But yes, they're clumps of widely spaced polyps on long stalks, and they appear to originate from one central core. So they form these mounds, and the corallites are round to an oval shape. So they almost could look like a mouth, or they could look like a bouquet of flowers. But as you see in the different photos too, some of them appear like closer together, the mound, and some look a little bit more spread apart, and you can see the base a little bit better. All right, so then we've got Pseudodiploria strigosa, our symmetrical brain coral. The family is Favidae, and the acronym is PSTER. They grow up to 6 foot, and they span 3 to 130 feet. So this one is very common here. And you may think of it as like your classic brain coral. So you see a lot of these actually are very susceptible as well here. We find them pretty early on at a lot of these sites, and they're actually quite easy to treat. And then lesions are quite noticeable on these guys. So they have symmetrical lines that are vertical to the base of the colony, and their ridges are quite rounded and sharp. So very distinct ridges and these very distinct lines. They often have little holes within the colony as well. So sometimes you'll see little creatures living in them, and you'll find the lines lead into these like holes within the colony. So yeah, these are definitely one of our favorites to treat. They'll span shallow and deep, and some of our sites have plenty of different brain corals that we'll treat. So the picture on the far top has like numerous lesions. So that probably would be, that one appears to be more than half dead. So that would be something that you wouldn't treat. The others are completely healthy. And you can see the one on the top left is a nice boulder coral, and the one on the bottom is more of like a platal spanning. So you've got Pseudodiploria clivosa. So same family, different species. It's a knobby brain coral. I call it creepy Uncle Clive because it looks really funky. So the family is Favidae, and the acronym is PCLI, PCLA. So they grow up to 4 feet, and they're 3 to 135 feet deep. They have all these irregular knobs oftentimes, and their lines are not parallel to each other. So if you see the picture to the top left, that is showing a bunch of the different knobs that it could appear like, and then at the bottom it's more of a smooth appearance, but you can still see these lines are not parallel to each other. It has like a funky look about it, but you can still see there are very distinct lines as there were with the Pseudodiploria clivosa. It's just a very different appearance than it is. So yeah, creepy Uncle Clive, but you can think of whatever ways you'd like to remember some of these corals. So Colophilia natans, this is our Boulder brain coral. So the family is Favidae, acronyms are CNAT. They grow up to 7 foot. So these ones get really large, and they can be 2 to 175 feet deep. So these are some of our priority ones to treat off of the wall. And as you get a bit deeper, you see some really large colonies of CNATs. And they're very beautiful, and so it's pretty devastating when you see some of these that are slowly dying. So, and just because you see such large colonies, they really are a priority for us to treat. They have big valleys in between their ridges, and their valleys often look green, but sometimes not, and you do find them in large colonies at times. The one at the bottom wouldn't be a large colony. You can't really tell from the picture, but that would just be a small section of CNAT, and the other two look like they'd be bigger colonies. The one on the top far right looks like it could be a huge one. That part of it is dead. Actually, that's a picture of someone treating a CNAT coral. So you see our measure bar in the side, just so that you're able to see the size of the coral a little bit better. And it looks as if SCTLD has actually infected part of the coral. So part of the living tissue is still alive that they're trying to protect there. So yeah, like I said before, it can appear green in between the valleys. So that's a good way of distinguishing this coral. But color is not always the best way to distinguish, because it may appear different, may not be green. So you may see something like the bottom picture, and you'll just have to go by more the shape of the coral. So the spacing in between the different ridges. Yeah. So Dlabs, Diploria labyrinthiformis is a grooved brain coral. Again, the family of Favidae, and the acronym is Dlab. So these ones will grow up to 4 feet, and they're 3 to 135 foot. So a lot of people have, when you're starting to identify corals, a lot of people have trouble identifying between the Pseudodiploria strigosa and Dlabs. But once you look for certain things, you can tell they're actually very different. So they have these deep and narrow valleys, and they have the most distinguishing factor, I think, is that they have 2 parallel ridges that run alongside each other. So it's not actually one ridge. If you look closely enough, you can see it's 2 parallel lines that like follow each other along the coral. And then you have these deep, narrow valleys. So that's the best way to identify them. But we don't see those usually as much as the peasters, but they are also common on the reefs. All right, so those were our highly susceptible corals, and then we're going to talk about our moderately susceptible corals. But we did, as Katie mentioned before, the first 4 that I showed were our most highly susceptible species of corals. And then the other ones would be maybe after SCTLD has been at a site for a little bit of time already. And this would be secondary, so after SCTLD has been at the site for a little bit longer, but it doesn't always follow that rule, but that's usually what we go by. So this is Siderastrea siderea, our massive starlet coral. The family is Siderastrea, and the acronym is SID. So they grow up to 6 feet, and they're 2 to 200 foot depth. So these ones can get quite large as well. So I know Katie was talking to you earlier about innies and outies. So to me, this one looks as if someone has stabbed it with a pencil over and over. So they have these pitted corallites. So the surface is covered with them, generally symmetrical round pitted corallites. And you'll find different shapes of these colonies. So it may appear like a boulder, but then some of the younger colonies may be smaller and encrusting. There's also different species, Siderastrea radians, but you don't need to distinguish between the two. But that one you'll often find like on the surface, on the like bottom of the seafloor, and you'll find like little colonies of that one as well. But yeah, the biggest distinguisher is just you have these little pitted corallites. So instead of polyps being out, they're faced in. And this one actually is quite difficult to treat, and it's really difficult. It's to distinguish if it's SCTLD sometimes. You have like this actually does pop up as one of the first corals that seem to be infected at certain areas as well. But the coral almost turns like a purpley color oftentimes. And then sometimes you'll just have these nice white lesions, or it'll be like a mixture of like white and purple all over the coral. So if for this one, if there was a small, nice lesion, that would be one that I would treat. But if you see like a bunch of different lesions around and there's it's purple, it's white, it's like very messy, you wouldn't bother treating this type of coral because it looks like the entire colony is almost infected, and you'd be putting little dots of antibiotics all over the entire thing, and it'd be really hard to distinguish where the infection starts and where it begins. So yeah, if there's a a very clear lesion and it's just started to become affected on these corals, then that's something that we can treat. Otherwise, we usually don't, because you can't be sure that it's going to be effective. So MCAVs, we talked about this a lot before. These are a fan favorite, Montessori cavernosa, the great star coral. So the family is Fabidae and their acronym is MCAV. So they can grow pretty large as well, up to 8 feet, and they span 6 to 300 foot. So their biggest distinguisher is that they have these really large polyps, and they're quite protruding. So I remember this coral because, again, a weird way, but I think it sounds like a spell, Montessori cavernosa, and I think of Harry Potter, and I think of like if you put a spell on a coral, it would have huge polyps, and that's just the way that I connect the two, which sounds crazy, but that's how I remember Montessori cavernosa. So they've made these polyps huge. So they can be 2 to 4 times bigger than the average polyp. And they're often found in large mounds, but they can be a variety of different colors. So they could be yellow, green, brown, gray, or even fluorescent red or orange. Some of these are so beautiful on the reefs when there are these fluorescent colors. It's really amazing to see. And sometimes you'll see them more encrusting as well, but you will see them mostly in the mounds. So as Katie said, these are quite difficult to treat. So they are almost like have quite the mucus layer, so they're quite slimy, and because of the uneven, like soft polyps, it can be really difficult to put antibiotics on those corals. So the easiest way that we found is if you can't secure the antibiotics nicely to right on the lesion line, to secure the antibiotics to the dead skeleton. So this would be like something that doesn't have any of the sloughing tissue on it, something that's completely dead where you can get a base and you can stick your antibiotics on there and spread it from there to reach the infected lesion or to reach the leading edge of the lesion. So this one can be a bit tricky, especially if you have a bit of a surge or if you've got a bit of a current going on. But these ones actually do appear to be some of the corals that seem to be taking the treatment the best. So they are definitely worth it. And again, it depends. Sometimes you'll see a MCAV with a nice lesion on the side, only one lesion, and you know that you can really like, that would be a great coral to treat. And then sometimes you see them, corals that look like completely ravaged and they've got like 5 different lesions going on. And you know that that coral is probably a lost cause, which is sad. But yeah, it's picking your battles. Sorry, one sec. Yeah, these ones can definitely be challenging. Sometimes the polyps do appear to be closer to the surface of the coral, and then those ones can be easier to treat. It just depends sometimes. So we've got our Orbicella annularis, our lobe star corals. These are Fabidae as well, and their acronyms are OAN. They grow up to 10 foot and they span 6 to 130 feet. You often see them in the shallows though. They're one that you see a lot of times when you're snorkeling at some of these different sites. And as you can see with the picture in the top left, you get these like huge colonies. So it looks like a bunch of different boulders all together in one. So they can be a little bit of a nightmare to treat sometimes, because you'll have infections everywhere, just a little bit on the corals. But because they have such small polyps and it's not an uneven surface, they're actually really they're quite easy to treat and they don't require much antibiotics. So you can stretch the antibiotics nicely across them, just because it doesn't require much at all, and you can smooth it out. And so it really wouldn't take you much to treat an entire area of those, but it is time. It takes a lot of time. But yes, they have dome-like tops and these very small polyps, as I mentioned, and they do usually have a mustard color. Okay, so the same family, different species. You've got Orbicella fabulata, our mountainous star coral. So these are OFAVs. They grow up to 18 foot and they span 6 to 130 feet. So these ones, I find that I see them more in the deeper waters, and they're really cool to look at. Most of the time you see them plating, and they form these really large colonies. Sometimes you see like a bunch of different skirts of them. So you see almost different layers plating, all like spanned out. And they're the same as the OANs, where they really don't require much antibiotics to treat. You can spread it right across. They also have these really small polyps. They just seem different from the OANs most of the time just because of the structure. If you look closely, you can see little differences between their polyps, but that can be quite difficult. So it's easiest to kind of distinguish between them via their structure. But yeah, these ones are great. You see a lot of them on the deeper sites, and especially some of the sites where there's been SCTLD for longer. These are some of the corals that we're really approaching and trying to treat. And they also have these, yeah, they often have like skirting edges when you see them plating like that. So I think of like fabulata fancy, they're wearing a skirt. It's always the way I remember them, but okay, and then you've got Orbicella franksii. So this is your Boulder star coral. So the acronym is OFRA. They are up to 3 feet, so these ones aren't too large, and they can be found 6 to 130 feet. So they grow in irregular mounds, and they have like different incrustations. They have a bunch of different scattered lumps, it appears, on top of them. And a lot of the times the lumps will have like white polyps at the top. So don't let this fool you. This isn't SCTLD. It's not an infection, even though it does appear like you have multiple different lesions all over this coral sometimes. That's just the way that this coral does appear. So yeah, it has very unevenly distributed corallites of unequal size and height. So it appears like lumpy for this reason a lot of the time. And you'll see them sometimes in boulder short form, but you can also see them encrusting, as you see in the picture below. So I think of this, I mean, similar to the other coral I was mentioning, but I think of this as freaky Uncle Frank, because he's all lumpy and crazy going on. So we've got Agaricea agaricites is our lettuce coral. So Agariciidae, AGA is the acronym. These grow up to 3 foot, and they span 3 to 240 feet. So you've got these thickly encrusting plates. They almost resemble lettuce, but then you can also have hemispherical structures with different heights. So you find these. You can find them in large colonies, but often you see just like smaller colonies of these guys. And you'll find like the front and the back look pretty similar a lot of the time. So it's like encrusting, but like outwards, if that made sense at all. But I don't tend to see these. Yeah, so there's a lot of different species in the Agaricea family, as Katie was mentioning before. Agaricea agaricites is just one of the more common ones, and it can be hard, especially when you're first learning your corals, to distinguish between some of them. So all that you need to know for now is that it's an Agaricea. These ones do take up a little bit of antibiotics because to get it in between all the different grooves, it takes more than you would think, especially when you have like these, even when you have smaller lesions. But these ones are one of the moderately susceptible. So we've been starting to see more of them get infected at some of our sites. But yeah, they're very distinguishable just from the structure of like these uneven ridges and funky grooves. It almost looks like little like pitted holes in between in different areas. Yeah, but they can appear in a number of different forms. So then you've got our Mycetophilia species. So the family is Mucidae, and there's a few different species of them. So yeah, the new one didn't come up. But you've got Ferox, you've got Lamarckiana, and what was the other one? Aliciae. Yeah, so there's, again, you don't need to know the few different species of those. It's good enough to know the family, Mycetophilia, for now. These, I think, look like quite the ancient species. You'll find them encrusting, and they have very spaced ridges. So some of them also appear to have like little dots in between in the valleys, but they do have this like kind of freaky look about them. You'll find them gray, sometimes you'll find them in like different colors, sometimes you'll find like a stark contrast between the ridges and the valleys. It may be like a bright green or something like that. And these ones with the lesions I've been finding is that it almost appears to be like purpley color on like the leading edge area, but that is still SCTLD. So sometimes it takes like a bit of investigative work to figure it out with some of these different species. And the more that you're diving, the more that you'll become comfortable recognizing SCTLD in some of these different species. That can be challenging at first. But yeah, these ones you don't see as many of them that are infected, but you do time to time. Okay. Well, that did fine. That one uploaded. So then we've got some of our low susceptibility species that Katie mentioned earlier. So these are ones that you wouldn't expect to be infected by SCTLD, or if you do, at a very low rate. So we've got our Porites species. So we have Porites asteroides, our mustard hill coral, which is also called past. So you see lots and lots of these around the reefs. It's exactly what it sounds. It looks like a mustard hill. So you'll often find these in like a mustard yellow color. You can find them on mounds. You can find them encrusting, and they just look like these lumpy formations. Because of their polyps, they do almost look like they have a fuzzy appearance. And it's the same with the other species of this family. So Porites are club-tip finger coral. They are, their acronym is PPOR, and they look like almost the fuzziest. They look like really fat, fuzzy fingers almost, and you'll find them in these big clusters sometimes. And that can be often difficult to distinguish between Porites furcata, which is the branching finger coral, but those tend to be less like clumped together. They tend to be more branching out, and they tend to be like a little bit thinner and less of like these stout kind of fuzzy fingers. And yeah, so for the Porites and the Porites furcata, we haven't seen any of these infected, luckily so far. The Porites asteroides, we've been seeing some things that do look like infection, but it's really difficult to tell, especially because other science has been saying that these are one of the, the less likely corals to be susceptible to SCTLD. So it is one we're watching out for and we're looking out for sure. And then we've got our Acropora species. So you have Acropora cervicornis, and we've got our Acropora palmata. So luckily we haven't seen that these species have been affected by SCTLD as of yet. As Katie mentioned before, especially the Acropora cervicornis are ones that are typically used in coral nurseries, or have been in the past a lot of the times. They're quite the branching coral. They're very easy to take fragments off of. So they're very fast growing. So they're really popular with these nurseries. And the Acropora palmata, both of them are really great reef building corals. So especially because these ones have been affected by different diseases in the past, and we've had die-offs of these species. We're really lucky so far that they don't seem to be being affected by SCTLD. Yeah, so that was a lot of coral ID knowledge, and you guys have gotten a lot of info over the past week, but it is great to start learning about your corals. It makes diving and snorkeling more interesting. It makes you more efficient here with the DoE, easier to know what you should be treating, what you shouldn't be treating. It makes it easier for you to communicate any questions that you have. But there are different resources for you to use, for you to study up more on your corals. So you've got the Agro website, you've got Coralpedia, and then also your coral identification books. So I've got one here. So you can find those online via ebook, and then you can also find them in certain bookstores, but they're so helpful. I'm always using my coral books. It makes it easy if you have a question about something, you see something underwater, you take a picture of it, you're not 100% sure what species it is. These have all of your Caribbean reef corals. So it's really easy, and you can learn about different distinguishers between them. You can learn all of the information that we shared and more. You can see a variety of pictures. And so, yeah, those are amazing references for you to use. But also, a lot of us here are pretty comfortable with our corals, so we're happy to talk to you guys about anything. But yeah, we encourage you to start trying to learn these things and to start paying attention when you're underwater. And start trying to learn your coral ID. So we have a Kahoot game for you guys if you would like to play. I don't know if you guys have played Kahoot before, but basically you open up the website on your phone and you type in Kahoot, and then I give you a code and we can all play together. It's quick, it's just 12 questions. It's about coral ID. You may know some of the answers, you may not, but it's just a bit of a challenge for you guys. Yeah. I see Cynthia, but I don't see anyone else. But yeah, do you want to play?
I don't have a phone.
Oh no, you can open it on your laptop.
Oh, okay. Well, that's what I'm using for you.
I'll show you guys. Okay. So I'll put it on, then we can go. Oops. Oh wait, don't look at that.
I think you need to exit the
Oh, no.
PowerPoint because you're still sharing the screen for the PowerPoint.
Oh really?
Mm.
Okay.